What does a sentence, a Lego tower, and your pancreas have in common? Well, they all depend on one thing: order. And in biology, that idea of order is absolutely critical, especially when it comes to proteins. From enzymes that digest to hormones like insulin, the function of a protein depends on the exact order of its building blocks. That order is called the primary structure and is the foundation of how proteins work. Proteins are incredibly complex molecules, and scientists describe them using four levels of structure, kind of like zooming in from the outside. First is the primary structure, the simple sequence of amino acids all strung together. Then comes the secondary structure, which involves spirals and folds called alpha helices and beta sheets. Next, we have our tertiary structure, where the whole chain folds up into a three-dimensional blob. And finally, the quaternary structure, where multiple proteins come together to form one large superprotein. But guess what? All of that fancy folding and teamwork starts with the primary structure. It is the blueprint. Now get that wrong, and the whole protein could fall apart. So what exactly is the primary structure? It is the linear sequence of amino acids in a protein. Basically, the order in which they're connected. You can think of amino acids like beads on a necklace or letters in a sentence. When you join them up in a specific order, you get meaning. These amino acids are linked together by peptide bonds, strong chemical bonds that connect the amino group of one amino acid into the carboxyl group of the next. And these proteins have direction. They're not symmetrical. They start at the N terminus, the N with a free amino group, and N at the C terminus, the N with a free carboxyl group. We always read protein sequences from N to C, kind of like reading left to right in English. Now, each amino acid in this chain is called a residue, and we number them based on their position on this chain. Glycine might be residue number 1, alanine number 2, and so on. And here's why that matters. If you change just one amino acid, just one, you can completely change the shape and function of the protein. It's like switching a single letter in a word. Cat becomes cut. Different meaning and different outcome. Now, before we move on, let us test your knowledge. Make sure to pause the video and answer the question at your own time. Let's test what you just learned. First question: what do we call the N of a protein that starts the sequence? Is it A, the Z terminus, B, the N terminus, C, the C terminus, and D, the front terminus? If you said B, N terminus, well, good job, you got that right. Now for our next question. Protein's primary structure refers to what? A, the 3D shape of the folded protein, B, a list of all the proteins in your body, C, the sequence of amino acids, or D, how the protein smells? Well, if you answered C, then you got the right answer, the sequence of amino acids, because that order is everything. Now, to get back to our topic, let's talk about why that order matters so much. Primary structure is not just a list of ingredients, it's a recipe. That exact sequence determines how the protein folds, how it behaves, and what it can do. If the order is off, even by one residue, it can have serious consequences. For example, sickle cell anemia. This disease is caused by a single amino acid substitution in the protein hemoglobin. One glutamic acid gets replaced by valine, and that small change causes the whole red blood cell to become misshapen, less flexible, harder to move, and worse at carrying oxygen, all because of one change in its primary structure. So, yeah, that sequence of amino acids is a big deal. Now, let's look at another real world protein, such as insulin. Now, insulin is a hormone that regulates our blood sugar, and it was actually the first protein scientists ever sequenced fully, thanks to the work of Frederick Sanger. Now, knowing a protein's primary structure, such as insulin, helps scientists understand what it does, diagnose problems when something goes wrong, and even design new therapies. In genetics and biotechnology, sequencing proteins tells us what the genes are activating. Now, in medicine, it helps us design drugs that can mimic or block certain proteins. And in synthetic biology, we can even engineer new proteins with totally custom primary structures. So, whether you're curing diseases or designing synthetic enzymes, the first step is knowing this sequence, its primary structure. To wrap up what we've learned today, we looked at how proteins aren't just blobs, but have structure. We also explored the hierarchical levels from the simple amino acid chain to complex folded forms. We also zoomed in on the primary structure, which is the linear sequence of amino acids that gives protein their identity. We also looked at the directionality from N terminus to C terminus and why changing just one residue can flip a protein's entire function. We also saw how the knowledge powers breakthroughs in medicine, genetics, and biotech. So the next time you hear someone say, it's just a simple sequence, you can say, actually, that sequence could change a life. So thank you for watching and joining us here in Catalyst Learning Center, and I hope I see you in the next lesson.